Hello everyone, welcome to today's live broadcast, Evaluation and Comparison of Non-Derivatization and Derivatization Tandem Mesh Spectrometry Methods for Analysis of Amino Acids, Acylcarnitines, and Succinylacetone in Dried Blood Spots. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. Thermo Fisher Scientific helps their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. For more information, please visit www.thermoscientific.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. Before I introduce today's speaker, we'd like to know a little about our audience today and ask you to respond to 3 short polling questions. Your help is very much appreciated, and we will use this information to help develop topics for future webinars. So our first question is, which kind of organization are you from? Please check the closest type of lab to yours. And A is Inborn Errors Metabolism Lab, B is Reference Lab, C, Clinical Research Lab, D, Forensic Toxicology Lab, E is University Hospital Lab, and F is Others. And I can just read that again. A is Inborn Errors Metabolism Lab, B is Reference Lab, C, Clinical Research Lab, D, Forensic Toxicology Lab, E, University Hospital Lab, and F is Others. Give you a minute to answer that poll. Okay. Our next poll asks, which technologies are your lab currently using? And you can select multiple choices here. So A is LC-MS, B is HPLC, C is GC-MS, D is immunoassay, E is biochemical technologies, F is PCR, and G is next generation sequencing. And remember, you can select multiple options here. Give you another minute to answer. Okay. And our next one is, which applications are your lab currently running? And again, you can select multiple choices. A is Inborn Errors Metabolism, B, Forensic Toxicology, C, Endocrinology Research, D, Therapeutic Drug Research, E, Protein Peptide Quantification, F is Others. And remember again, you can select multiple options. Give me a moment to answer here. Okay, so thank you so much. So your speaker today is Joe Bussolo, PhD, Senior Applications Scientist, Thermo Fisher Scientific Incorporated. Joe Bussolo earned his DS degree in clinical chemistry at Westchester University in 1977. In 1982, he received a PhD degree in biological sciences from Drexel University in Philadelphia. During those college years, Joe worked part-time as a medical laboratory technologist at Children's Hospital of Philadelphia. For most of the 20 years that followed, Dr. Bussolo helped develop and supported various analytical instruments and software products for the Perkin-Elmer Corporation. In 2002, Joe joined Cohesive Technologies as an applications scientist. Cohesive was acquired in 2007 by Thermo Fisher Scientific, and Joe currently works under the Thermo umbrella as a senior scientist, training and collaborating with customers and managing the applications laboratory at Westchester University of Pennsylvania. He is also an honorary faculty member and advisor in the university's pharmaceutical product development program. I will now turn it over to Joe for his presentation.
Hello, and thank you for joining our webinar on analyzing dried blood spots for researching inborn errors of metabolism. I'll be showing you work done by applications scientist Cheryl Z under the direction of our applications manager, Marta Kozak. This presentation will help you decide whether or not it is necessary to derivatize dried blood spot extracts in order to measure various amino acids, acylcarnitines, and succinylacetone by Tandem mass spectrometry. We will first go through the technology that allows us to make such measurements. We will then describe how a software product called IRC Pro can process the data from each specimen in order to study various inborn errors of metabolism. Finally, we will cover the advantages and disadvantages of using derivatization in preparing dried blood spots for such applications. So let's start with mass spec technology. New developments in electrospray ionization and tandem mass spectrometry during the 1980s and 1990s allowed the development of rapid, high-throughput analysis of samples extracted from dried blood spots. During this time, Professor David Millington and his associates at Duke University Medical Center used tandem mass spectrometry to investigate defects in enzymatic oxidation of fatty acids in mitochondria. Such defects led to the accumulation of acyl coenzyme A intermediates that eventually leave the mitochondria as acylcarnitines. The Duke University researchers figured out how to extract dried blood spots and how to use tandem mass spectrometry to quickly analyze these compounds by flow injection without using chromatography. They eventually used tandem mass spectrometry targeted to detect several amino acids at the same time as they can detect 20 acylcarnitines in order to study over 30 metabolic diseases, including phenylketonuria, or PKU. And here we show in the bottom center of this slide an overview of one of our newest triple quadrupole mass spectrometers, which was used in this study. The samples, analytes, first get ionized in the source. Specific precursor ions are isolated in the first quadrupole. That's the Q1 mass filter, and selectively sent to the second Q2 collision cell, where they collide with argon or nitrogen to form fragments. These product ions are then isolated in the third quadrupole, which is the Q3 mass filter, and selectively sent to the detector. Specific transitions from precursor to product ions are recorded and presented by the mass specs software, enabling us to measure how much of each transition was formed from our sample. By mixing our sample with a set of stable isotopes of our analytes of interest, the system can measure the transitions of those as well as the sample analytes themselves. And that allows us to estimate the amounts of each analyte in our sample. This technique is known as isotope dilution. Okay. The compounds listed here include amino acids, succinylacetone, that's SUAC, and then there's carnitine itself, which is a C0, and the various small chain, that's C2 to C4, and medium chain, C5 to C8, and then there are the long chain and very long chain, that's the C10 to C18. Those are the acylcarnitines, and they get extracted from the dried blood spots. The Tandem mass spectrometry system can measure these individual sample analytes with high selectivity and sensitivity, enabling us to detect and quantify multiple analytes from a single injection into a flowing stream, even without derivatization. The workflow for doing such analysis is illustrated here. Dried blood spots are punched, and the resulting discs are extracted with a buffer containing internal standards. This part of the process can be automated to utilize 96-well microtiter plates. The plates are then placed in an LC system that automatically injects extracts into a stream of solvent flowing to the mass spectrometer. In this case, the LC system does not utilize an LC column. Instead, we do flow injection analysis. So with each injection, the mass spectrometer applies ionization source conditions, as well as mass filtering and collision-induced dissociation conditions that have been optimized for each analyte and its internal standard, so that selective monitoring for each is recorded and processed. In addition to the software used to control the hardware and process the raw data from the mass spectrometer in a generic fashion, we have a special software tool called IRC Pro that you see over on your right, which takes that raw data and automatically quantitates analyte concentrations and performs custom calculations such as analyte ratios. In order for analytes to be analyzed by a mass spectrometer, they must be ionized. The sensitivity of any mass spec method is proportional to how many ions are formed and how many of those ions get into the mass spec. Some analytes are already ionized with just one charge and are easy to deal with. Others are not. Amino acids and acylcarnitines are zwitterions. They have both positive and negative charges that sort of cancel each other out, typically at pH values from 4 to 8. If we lower the pH enough, we can protonate the carboxylic acid moiety of these molecules to make them positively charged. We can also chemically modify these molecules using butanol and HCl, which converts carboxylic acids to butyl esters. This derivatization strategy ensures that all of the analyte molecules are positively charged in the mass spec source, and a larger percentage of them are drawn into the quadrupoles. Earlier models of mass spectrometers lack the sensitivity to reliably measure small amounts of such analytes unless they are derivatized. So as we will see later in this presentation, the Endura's sensitivity is so good for amino acids and acylcarnitines that derivatization is not necessary. The triple quad mass spectrometer can selectively detect and measure derivatized acylcarnitines and amino acids by 3 different scan modes. At the top you see the neutral loss scan looks for amino acids such as the phenylalanine shown here that lose a fragment with a specific mass of 102, and that's actually butyl formate, as you see there. The precursor ion scan next looks for acylcarnitines that produce a fragment with a specific mass to charge ratio of 85. We also use SRM scans, that's selectively or selected reaction monitoring, that look for specific precursor ions that form specific product ions for both derivatized and non-derivatized analytes. The TSQ Endura quadrupole mass spectrometer has a unique design to achieve the best sensitivity for all of the scan modes we just mentioned for derivatized and non-derivatized analytes. If we look at the ion optics, we have RF lens followed by a bended ion beam guide with a neutral blocker. And then we have hyperbolic quadrupole mass filter for Q1 as well as Q3. And separating Q1 and Q3 is a curved collision cell. All these combine to reduce noise and increase sensitivity for enhanced quantitative performance. This is especially important when introducing samples that have not gone through extensive cleanup. Ultra-fast selected reaction monitoring, or SRM, as high as 500 SRMs per second, with up to 30,000 definable SRMs in our acquisition method. This enables quantification of more compounds in shorter time frames. And then at the end here, you see a 5-order magnitude dynamic range of the dyno detector, which increases quantitative confidence. So with this unique design, we believe we do have the best in-class sensitivity with wonderful usability and, of course, robustness that's needed for this kind of application. The unique design of our mass filters, which utilize hyperbolic quadrupoles, and the unique way the radio frequency, or RF, is applied to them, result in a highly efficient transmission of ions even at high mass resolution. And there are times when sample matrix interferences are evident in flow injection methods, and since chromatography is not used to resolve those interferences, switching the mass spec to high resolution filtering for the precursor ions becomes critical to achieve selective detection and quantitation and eliminating the influence of any interferences. The unique design of the curved collision cell enables SRM scan speeds of up to 500 SRMs per second with highly efficient ion transmission. If you are using a mass spec with ultra-high performance liquid chromatography, you get very sharp peaks, and you need to get at least 7 points across the sharpest chromatographic peaks in order to integrate them properly. However, if your scan limit is 200 SRMs per second, which is the case in most triple quads, you are limited by how many transitions you can measure simultaneously. If you don't use an LC column and do flow injection analysis, as we do for acylcarnitines, then this performance becomes very necessary, in fact critical. If you don't have time or resources to do extensive sample cleanup, then you risk contaminating the quadrupoles in the mass spectrometer. The data that you see here is from making multiple injections of alprolizam in synthetic serum. Our novel ion optics design stops neutral molecules from our sample from getting into the quadrupoles, thereby increasing system uptime by 3 times over previous designs. This level of reliability is especially important for applications that utilize flow injection analysis without chromatography. The next topic that we should cover is data processing. In addition to software used to control the hardware of our LC-MS system, which also collects the raw data, processes it, and presents generic qualitative and quantitative results, we used IRC Pro software to analyze the results in more detail. As you know, errors happen, both random and systematic. Following standard operating procedures and utilizing hardware and software tools to uncover and minimize errors must be a priority in every laboratory. I will describe a software product that reliably copies the data from our LC-MS system and performs the necessary calculations and generates reports for interpreting the results. That software is called IRC Pro, which is made for research use only. As data is acquired by our LC-MS system, IRC Pro copies it and does its own peak area integration. Here we see a peak that was formed for the SRM transition 209 to 107 from a flow injection analysis of a specimen. The area under the curve is calculated by specifying a percentile of the maximum intensity so that only the intensities above the noise level of the baseline get integrated. So we're able to integrate this part very accurately and ignore the rest, which is attributed to noise. After determining the peak areas for analyte using its SRM signals and for its internal standard, the software calculates the concentration using a relative response factor for that analyte. The IRC Pro software can do custom calculations, such as adding and subtracting and doing all sorts of ratios. For instance, Formula 1 is used to investigate tyrosinemia types 1, 2, and 3. Formula 2 is used to research medium-chain acyl-CoA dehydrogenase deficiency. And Formula 3 is used to investigate carnitine uptake defect. And the IRC Pro can do all these calculations quite easily. IRC Pro is easy to use. First, we program the settings, defining analytes, analyte types, and sample types. We then set up the method, which defines mathematical formulas and concentrations. Finally, we process the mass spec data from LC or flow injection analysis experiments as they become available. If our analysis involves 19 compounds, which include 10 sample analytes and 9 internal standards, and we are to run a batch of 480 samples, IRC Pro does the thousands of calculations automatically without transcription errors. To validate the IRC Pro method, we compared its results to generic results from the software used to control the LC-MS system for 9 analytes from 480 specimens. We see excellent correlation of the IRC Pro results on the x-axis with the generic results on the y-axis. The residual plots for both peak areas and concentrations showed acceptable dispersions or variances. So the variations here are nice and tight. The formulas that were automatically done by IRC Pro were also validated by comparing those results to manual calculations done from spreadsheets, which typically take a lot of time and effort to do. Again, we get good correlations with acceptable dispersions or variances. These are typical validation results of our IRC Pro method that processed results from thousands of injections of specimens containing the analytes shown here. Out of 9,120 peak areas, almost 88%, that would be this part here, had a bias of less than 5%. Out of 4,800 concentration calculations, almost 85% had a bias of less than 5%. The custom formula that you see here had a bias of less than 5% for all 480 calculations. A more complicated formula that you see down here involving amino acids and acylcarnitines, that had a bias of less than 10% for 81% of the 960 calculations that were performed. So this formula stresses the system, and I really don't know many others who would use such a complicated formula, but this was done to show you how we're able to handle very complicated formulas by IRC Pro. After covering the instrumentation technology and software used to acquire and process data, we can now focus on sample preparation. The question is, do we need to derivatize or not? For this study, amino acids, acylcarnitines, and succinylacetone were extracted from dried blood spot punches using a single extraction step similar to that of Dillon et al., involving acetonitrile with 20% water and 0.05% formic acid. The derivatized approach involved butylation of the carboxylic acid moieties of each compound, which takes an extra 45 minutes for the reaction and evaporation of the butylation reagent. This is the LC-MS system used to analyze the prepared samples using flow injection without chromatography. The system consisted of an ultimate binary UHPLC pump and an open Acela autosampler for flow injections to the heated electrospray ionization source of a TSQ Endura Tandem mass spectrometer. And of course, all of these are from Thermo Fisher Scientific. The Endura's mass spec conditions you see here were optimized for the amino acids, succinylacetone, carnitine, and acylcarnitines. Also shown are the flow injection parameters, as we injected 10 microliters of sample into a flow of acidified water and acetonitrile solvent at 90 microliters a minute. The analysis time for each injection was at 1.5 minutes. So this permits a throughput of almost 40 samples per hour. These are some of the analytes and internal standards that were used in the study with their SRM transitions and optimized mass spec parameters, that is, collision energy and RF voltage, to get the best signals for the butylated derivatives. There were also 12 amino acids, succinylacetone, carnitine, and 18 acylcarnitines in each sample. These are the corresponding parameters for non-derivatized analytes and internal standards. Represented peaks from flow injection analysis detected by SRM transitions for succinylacetone, that's SUAC, and glutaryl-carnitine, that's the C5DC, so that really is a 5-carbon dicarboxylic acid. So along with their internal standards, you can see that the detector's response for the butylated SUAC was over 30 times greater than that for the non-derivatized SUAC. Derivatizing the C5DC compounds increased response factor by around 5. However, the detector responses for the non-derivatized compounds were more than enough for reliable quantitation. Also notice the runtime of only 1.5 minutes per injection. Well, this is flow injection analysis, no chromatography needed, which means no equilibration, no column washing. So this is very, very quick. We achieve a throughput of 40 injections per hour. One question you may have is, what is the difference in lower limit of quantitation between derivatized and non-derivatized amino acids, as well as the other compounds? These methods are semi-quantitative and do not involve calibration curves or LOQ values. The concentration of each analyte is estimated from its peak area ratioed against its internal standard peak area, and then multiplied by a response factor. For the quality control specimens, low, medium, and high, and these specimens actually come for the Centers for Disease Control, here we see for a derivatized sample set, within run precision was excellent, and run-to-run precision was very good. The concentration ranges in micromoles per liter of these amino acids ranged from 65 to 290, that's micromoles per liter, for the low QC, 150 to 400 for the medium QC, 250 to 640 for the high QC. And the ranges for the acylcarnitines were from 0.2 to 5 for the low QC, 0.5 to 10 for the medium QC, and 1 0.0 to 15 in the high QC. So that gives you an idea of what kind of response factors that we're getting. The non-derivatized sample set also showed excellent within-run precision and good run-to-run precision. In fact, if you look at the precision numbers, if you just go back and forth for some of the compounds, precision numbers for the non-derivatized batch are often better than those for the derivatized batch. And that's something that does happen when you do derivatization, because you're adding an extra step to the method. Even though we're getting better sensitivity, we're adding an extra step, and therefore we can add more variance to our measurements. So that's not at all unusual. Looking at the average precision of all 12 amino acids and 18 acylcarnitines, we see that differences between derivatized and non-derivatized sample sets were not significantly different from each other. These precision values were very similar to those achieved by Metz and others from derivatized sample sets. Comparing derivatized and non-derivatized results, we see excellent correlation for the amino acids and acylcarnitine results, even at low concentrations, where derivatization is necessary if you're using a less sensitive mass spec. The concentration ranges of many amino acids were above 100 micromoles per liter, while those for the acylcarnitines were less than 1 micromole per liter. In conclusion, the flow injection analysis of dried blood spots for measuring various amino acids, acylcarnitines, and succinylacetone can be done reliably without derivatization using the TSQ Endura Tandem mass spectrometer. Also, our software tool IRC Pro improves the time effectiveness by eliminating manual calculation processes and removing transcription errors. The method differences between quantitative values resulting from the two methods, derivatized versus non-derivatized, were minor. So it does show very clearly that using the highly sensitive TSQ Endura, we don't have to derivatize. We thank you very much for attending this webinar, and we can now open this up for questions and answer.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Dr. Xiaolei Si is an application scientist from Thermo Fisher Scientific who did the research work for this presentation. He will be joining Dr. Bussolo to answer your questions. And our first question is, do two quadrupole mass filters Q1 and Q3 have the same polarity or alternated? I think this question is for Dr. C.
Okay, for this question, actually for either Q1 or Q3 quadrupole or mass filter, so each one basically has 4 rows, and these 4 rows actually have 2 pairs of the polarity. Positive and negative, and then so that means so each quadrupole has these 2 polarities.
Okay, and we'll be asking some more poll questions during the Q&A. And so our first poll question is your lab running an inborn errors metabolism application? And your choices are yes and no. Give me a moment to answer that, and we'll move on to our next question. Again, that question is your lab running an inborn errors metabolism application? Yes or no? Okay. Our next question in the Q&A is to compare the pros and cons of the 2 methods. And we'll pose that question to Dr. C.
As we can see from the presentation, both methods are highly correlated. There are differences between these two methods though. So basically, derivatization method requires a lengthy derivatized process and utilizes environmentally unfriendly chemicals. Compared to derivatized methods, non-derivatization method is rapid and cost-effective. However, derivatization methods generate cleaner samples and high signal-to-noise for analyte of interest. So it's really up to the users to choose which method to use in their lab.
Okay, and we're going to ask another poll question right now, and that is, which topic do you want to hear from us in the future? First is forensic toxicology, endocrinology research, therapeutic drug research, multi-channel LC, for example, Prelude, TLX, etc., or software, TraceFinder, ToxFinder, etc. And I'll give you a moment to answer that if we move into our next question. Okay, and our next question is, how much difference of the signal, how much difference of the signal when comparing derivatization to non-derivatization methods? I believe this question is also for you, Dr. C.
Yeah, depending on the analytes, derivatization method increases the signal by anywhere from 3 to 40 times compared to non-derivatized methods. Derivatization methods would be the better choice if you have a mass spectrometer that is not as sensitive as some of the newer mass spectrometers. The thermoscientific TSQ Endura MS is sensitive enough for either derivatization or non-derivatization methods.
Okay. And we have one more poll question, and then we'll be answering more of your questions. The last poll question is, do you think today's webinar is helpful? And the choices are yes and no. Give me a moment to answer that, and then we'll move on to our next question. Okay. Next poll question is, have you tried using an automated DBS system, e.g. the DBS-MS 500 from FIA-MAG for the direct flow injection?
No, we haven't tried this automated DBS system. So what we did for this presentation is kind of using manual sample processing.
Okay. Next question, how can non-derivatization direct injection permit high analytical specificity for complex sample analysis? So derivatization sample preparation does give us basically a little better specificity because it will increase the mass of the analyte. However, the non-derivatized method can also achieve pretty good specificity, although it cannot differentiate some analytes with the same mass, such as the C3DC and the C4 hydroxy C4. But actually, in practice, somebody can use some curve
to make some calculation between these ratios of these two analytes, and can still achieve the practical needs. So I could argue that both methods can be used and generate pretty reliable results.
Okay, thank you. Next question is, how about the method correlation for any specific analyte?
So minor differences, one of them is less than 15%, were observed for the majority of the analytes between the two methods. But we do have the correlation graph for each individual analyte we monitored, but we just don't have so much room to show in this presentation. So if you want to say the correlation for any specific analyte, please leave your email address and let us know. We can certainly share the data with you.
Okay, thank you. Next question is, can TSQ access be used instead of Endura?
Yeah, the TSQ Access is a much more affordable triple quadrupole mass spectrometer from Thermo Fisher Scientific. And we've actually tested the derivatization method on the TSQ Access, and all of the amino acids and the succinyl compounds and acylcarnitines from dried blood spots can be detected and quantified using the derivatization method on the Access. However, the Access is not quite sensitive enough to handle the non-derivatized method.
Okay, thank you. And another question, how many samples can IRC Pro process at a time?
So there's no limitation in terms of the number of samples processed by IRC Pro software. Usually it takes 3 to 5 seconds to process one sample, depending on the complexity of the calculation. But if you want to process hundreds of samples at a time, so you better to run the software in a very fast computer with high capacity of the CPU and the RAM.
Thank you. We may have time for just one more question. Let me pose this one. Can IRC Pro be used for processing raw data acquired from other vendor instruments?
No, IRC Pro can only process the data acquired from a Thermo Scientific TSQ triple quadrupole mass spectrometer.
Okay, thank you. Another one, is IRC Pro open source?
No, the IRC Pro is not open source software.
Okay, thank you. Let's see. We can give our audience a moment to pose more questions if they would like to. Otherwise, we may close out. You see more questions may be coming in, so I'm just going to give you the opportunity, if you would like, to ask another question, and we'll just give you that moment. Otherwise, we will close out for today. Okay, you know what? I think we will close today, and you will have another opportunity to get more information. I want to thank the audience for their interesting questions and participation in today's event. We will respond in writing to questions we did not get to today. I would also like to thank our sponsor, Thermo Fisher Scientific, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through August 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.